Simone Guimaraes. Dr. Guimaraes has a bachelor's in veterinary medicine at the Federal Rural University of Amazonia, a master's in veterinary medicine at the Veterinary School, and a PhD in animal science at the Federal University of Minas Gerais. She held postdoctoral positions at the Iowa State University, working with molecular genetics applied to swine meat production, and at Wageningen University and Research in partnership with the Institute for Pig Genetics, working with swine genomic selection. Currently, she's a full professor at the Department of Animal Science at the Federal University of Viçosa in Brazil. Dr. Guimaraes has experience in genetics, focusing on genomic selection, genome-wide association, proteomic, post-genomic, and functional genomics. She supervised and co-supervised more than 120 master's and PhD students and published more than 230 papers and 300 conference abstracts. So thank you for joining us. Dr. Guimaraes, I'll give the floor over to you.
Okay, thank you, Kristen. So first of all, I would like to thank you for the invitation. It is an honor for me being here with you today. And thank you also for our colleagues that are here today sharing with us this moment, okay? So getting started, today we're gonna talk a little bit about what our team is doing on genomic studies in dairy Zebu and its crossbreds, okay? I will just talk a little bit about what we are doing. Of course, there are many other teams working with genomic studies in Zebu, okay, in Brazil, and even in our university and other universities in my state, okay? So it's something that is being fully studied currently in these days, okay, the genome of Zebu animals. So first of all, let me tell you where we are. So where is Viçosa? We are in Brazil. Brazil, I think you all know, we are in South America. So United States, Mexico, and Brazil. Okay, so in South America, we are in Brazil, and Minas Gerais state is here in the central part of Brazil. So we are very close to Rio de Janeiro. I bet everybody already heard about Rio de Janeiro. So by car, Viçosa we are here, Viçosa, if we have the map of Minas Gerais state. So we are about 5 hours from Rio de Janeiro, okay? So our university, this is the main building of our university. So I have seen that we have students from Viçosa in our audience today. So I think many people there will get a little bit, well, we remember about Viçosa now, seeing these pictures, okay? We were launched in 1926 as a land-grant university. Our model was the University of Florida, so 100 years ago, when the president of Brazil, that is native from here, from Viçosa, Dr. Arthur Bernardes, he invited from the University of Florida, Professor Peter Henry Hofs, to come to Viçosa, okay, to set up a land-grant university. And the objective at that time, 100 years ago, was a university focused in environment, animal science, and agriculture, because at that time, the Brazilian government thought about being in Brazil a big place for food production. So actually, I think that at least this objective, we can say that we reached it, okay? But nowadays, we are a more comprehensive university. We have more than 30 undergrad programs in our university. So it's a very nice, a very beautiful university. Just an example, 100 years ago, this was the house where Professor Peter Henry Hofs lived when he came from Florida to Viçosa. Nowadays, the same building, we try to maintain, we try to preserve our historical buildings. And this building nowadays is where our rector works. So it's fully conserved, okay? And the ranking that we present nowadays at the Times Higher Education for Agriculture and Forests as well, we are ranked. In the Animal Science Department, that is the place where I work mostly. This is a picture, and if you want to know a little bit more about the Animal Science Department, here is our homepage. We have a page in English, so you can follow it, okay? It's the most traditional graduate and postgraduate programs in Latin America. So the department was launched in 1926 with the university. So we are the oldest animal science department in Latin America. Our master began in 1972, and our PhD program began in 1976. So we are the oldest master program in Brazil, considering all areas. Okay, considering all areas, we are the oldest master program, all areas in agriculture and animal science. Okay, we are the oldest in Brazil. So far, we have already defended more than 1,200 master dissertations and more than 600 PhD theses during all these years, okay? And through the years, the focus of our department is being animal production in tropical environment. We are a tropical country mostly. We have a lot of subtropical areas as well. So we need to be focused on how can we improve production using this tropical environment. So we develop research on breeding, forage, nutrition, microbiology, behavior, physiology, reproduction, focusing on these tropical environments. And here is the picture only for cattle, but we work not only with dairy, but mostly with beef, pigs, chicken, and other animals, okay? But focusing on the adaptation of these cultures to the tropical environment. We also have European animals like Jersey and mostly Holstein, because we also studied how to adapt these genotypes, these taurine genotypes to the tropical environment. Okay, so we also have herds, for example, of the Holstein breeds here in our department. So what about milk production? Milk production for us is a challenge as a tropical country. The most important breeds are not well adapted to our environment. Holstein, Jersey, okay? So we need to know how to work with our environment and improve our production. If we take a look on this graph from FAO, we can see that South America doesn't produce too much milk when we compare to North America, Europe, and Asia. But we have a huge population. So we have to know how to deal with these problems about our climate and about the type of the forage that we use and improve our milk production. Okay, so for example, even in Brazil, we import milk from Argentina, Uruguay, and other places because we still not producing enough milk for our population. Okay, and we are being successful. This is the milk production in Brazil for the last 2 decades. So we can see that we are growing up. We still need to work on this growing development of milk production, despite the fact that we are in a tropical country. And Minas Gerais, that is the state where the Sosa is located, is the main state producer of milk. So we are responsible almost for 30% of the Brazilian production of milk. And what is interesting is that we use a lot of Zebu genetics and a lot of pasture, production under pasture. We also have some very good herds, some very good farms. Even here in Minas Gerais state, or more to the south, that use freestalls and hosting animals. We also have this type of production. But for the large majority of milk produced in our country, we're gonna have some crossbreeding with Zebu and the production under pasture, okay? So this is a very common type of crossbred in our country when we cross Holstein and Gyr. Then we generate different blood degrees of Holstein and Gyr. Okay, F1s or 3/4, 5/80 or 7/80. Okay, so this varies a lot, the level of hosting that we use in our crossbreds. When we do that, what we try to do is to put together the rusticity of the Zebu animals and the production from the hosting animals. Okay, and then we have a new crossbred breed that is called Girolando. We're gonna talk about it today, okay? Just an observation, this is beginning quite common in Brazil when you have the crossbred cows to produce milk and they are inseminated with beef breeds such as Angus. So our producers, they can make money not only with the milk, but also with the meat for the beef breed that was inseminated. So this is beginning to be common in our environment as well, okay? There are more than one breeds that are used for milk in the Zebu type of animals, but we're gonna focus just on the Gyr breed. This is the most important Zebu dairy breed. Here we have two pictures of two beautiful cows. If we compare to Holstein, we're gonna see that it's not a higher producer animal, okay? But we can say that under stressful environment, under pastures and low quality forage and low quality ration, we're gonna have the milking for 5,000 kilograms in 300 days of milking, okay? It's very nice because it adapts quite well to pasture. It's resistant to ticks and to hot environments. We're gonna show you some papers that we have developed in these matters. And what is nice is that we have official breeding programs that are, let's say, when we compare to the European breeds, we can say that our breeding programs for the Zebu breeds are at the beginning. But what is nice is that despite these programs are at the beginning, we are already including genomics. I'm gonna show you some examples that we can select our animals, our cows and the bulls by genomics. So we are beginning in good shape, let's say. Okay, this is from the Gyr Sire Summary 2021. The Sire Summary is the Embrapa, our company for Brazilian development and research in agriculture. They are responsible to generate this Sire Summary for our breeds, okay? But these Sire Summaries, they have the collaboration of several universities, including our university. So we cooperate with Embrapa on these Sire Summaries, okay? And here we can see the evolution of milk production at 305 days for the Gyr breed. So for example, when we began the breeding program, the average production for the breed was 2,000 kilograms for 305 days of milking. Nowadays, we more than double that, okay? We are approaching 5,000 kilograms per lactation, okay? So compared to Holstein, that's not much, but we can say that we are doing progress, okay? And I'm gonna show you about something about genetic variance that we have in Gyr also today, later in my talk. And then we can see that we still have room for more selection. This is a file from a farm where we have, I'm sorry, it's in Portuguese, I just didn't translate because this is the original file that came from the farm. This farm, it's a very important farm. This is like, let's say, a nucleus farm for gear production. So this farm, as well as other farms here at Minas Gerais state, they use only embryo transfer. They work with genomics, and we say that it's like a nucleus farm in the same, more or less in the same models as we have for pigs, because this farm, they have the most important lines, the purebred lines, purebred gear animals. And this farm, as well as other farms, they spread the gear genetics in our country, okay? And something that is quite nice is that despite the low production, casein, the beta casein in our gear animals, it's naturally A2A2. So it's a quite valuable milk, and the fact that we have A2A2 genotype at the gear breed, it's very frequent. It's the most frequent genotype for beta casein. This adds value to the milk of the gear animals, okay? And here you can see that all the cows that are being analyzed here in this farm, we have the genomic PTA for milk production, okay? So this is actually being used in the field, even for the Zebu breeds. Well, the Girolando breed is the crossbred between Holstein and Gear. So nobody knows exactly when these crossings began, okay? People used it to do that decades ago, let's say empirically. And what people saw in the field, in the farms, was that the cows, the crossbred cows, they were resistant to the environment, resistant to the poor quality of the pasture, but still having good production. Okay, so 1996, Girolando became, let's say, an official breed. So our technicians that just thought that it was so successful and it could be an answer for the challenge of producing milk in the tropics. So, okay, let's make these animals a breed, let's study it, and let's control the crossing and the breeding. So as I showed you some slides ago, there are several blood types that are allowed inside the Girolando breed. It's not, the book, the breeding book for Girolando is not closed. So we have an influx of genes come from Holstein and coming from Gear every year. We have breeding program that is also at the beginning. It's even newer than the breeding program for Gear. 80% of the Brazilian milk production come from Girolando animals. One of the blood types that we have for Girolando animals. It will be included here in 80% of the milk we produce. So you can see how important these animals are for our economy. They still can produce fine under pasture. They are resistant to ticks and hot environments, and they are also fertile. So it's, let's say, good business working with Girolando animals. These cows, they are, let's say, expensive animals because they produce more than Gear, and they are more resistant than the Holstein, and they can produce under pasture. Okay, so here are some data from the Girolando sire summary 2 years ago. 5,000 kilograms in average. Days in lactation. This is something that deserves research in the Zebu animals. The persistence of lactation. We still have to work because they're not too persistent in lactation. So we need to keep working on that to make the persistence higher, such as we have in taurine breeds. But the fat percentage is quite good because it follows more or less the pattern of the Gear animals. Age at first parity, 2.8 years, okay? But we have already seen that we can make this more precocious. But what happens many times is that as the producers want to keep these animals under pasture, they are not really very interested in making them too precocious, because this could mean that they have to put these animals in a freestyle, for example. So they prefer to have animals that are not that precocious in regarding age of puberty, but they can raise the animals under pasture. Okay? So this was the first paper that my team published working with genomics and gear, the dairy Zebu breed. So it was 2009. So it was, let's say, how many years ago? 15, 16 years ago, when we're looking for QTL on the chromosome 6 for the gear breed. Okay, so we developed a daughter design. So we had 14 families, the number of daughters. We found some interesting results. For example, here is we found a very nice QTL for fat yield, for fat percentage on the chromosome 6. Okay, the arrows means the microsatellite markers. So we had something very interesting at the genomic level for fat percentage. And when we disentangled the families that we had, we could see that family 4 had this QTL in its genome. So it came from the bull number 4. There was like an animal in our, let's say, nucleus herd. But what was the problem? Here we were working with microsatellites. Especially for the young people that are in the room, I'm not sure if you know what is that, but you do not have dense markers, a dense map, so it's quite hard to reach the quantitative trait nucleotide. Okay, so we just kept this in the drawer, looking for a better opportunity to develop more research on that. This was another paper that we published in 2010. This was for Girolando, the first one for Gir, and this was for Girolando. And we were looking for QTL affecting resistance to ticks. And it was quite interesting because we could analyze the same animals for tick resistance in the rainy season and in the dry season. And we could see that we had different results if the animals were in the dry season and in the rainy season. But it was also microsatellites, quite hard to keep working on that. So a few years ago, we could pick up the same population of Girolando animals. This is half-blood Girolando population, and work with the same population for tick resistance using SNPs, using the high density SNP chip. For the high density, the 700K, what is the problem is that in general, we can use around 420,000 SNPs, because the 700K, it was developed for taurine animals, okay? So in general, we can use a little bit more than half of the SNP chip information, but we still making progress. We already have some SNP chips that are developed for Zebu. And this is not as dense as the 700K, but this is specific SNP chips for Zebu already helps us a lot. So here we could make another mapping, looking for the resistance to ticks in this F1 population. And we could also develop another study here, that is the breeding of origin of the alliros. So we used a model that was at the very beginning developed for pigs. I have one of my foods is in pig genome. So I took some models that our students were using for pigs, okay, to trace the haplotypes in the crossbred pigs. And we adapted this model to be used in 2 breeds crossbred. And then we could use this for the Girolando and look for the origin of the alliros. And what we could see was that when we have the QTLs, we have the windows, and we have the significant SNPs inside each window. We could see the origin of those SNPs. So, for example, in the resistant animals, we could see that, for example, for SNP1, 38.2% of the SNPs for the resistant animals for SNP1 came from gear. Only 11.8% came from hosting in homozygotes. Okay, here we didn't study a lot about the heterozygotes, so let's focus just on homozygotes. For SNP2, more or less the same. 12% of the animals had were homozygotes for hosting alliros, and 39.4% were homozygous for the gear alliros. And you can see that for all the other SNPs, this tendency is the same. So we have among the resistant animals, more animals that are homozygous for the gear alleles than homozygous for the hosting allele. So this is more or less what was expected, because gear is more resistant. When we have this more susceptible animals, the situation is on the other side. Okay, we have more animals bearing homozygous from the hosting breed when compared to the gear breed. Okay. Well, so we thought that this brought for us interesting information. And then we decided to work with heat stress response. We had also phenotype for heat stress response. Let me see. Okay, yes, for heat stress response. So what we did was we submitted the Girolando, F1 Girolando animals. We submitted them to climatic chambers. The animals were some hours at a good temperature for them, a comfort temperature for them, and then they were submitted to a stressful temperature. And we saw the response of the animals. So we made that for around almost 500 animals. So it was a very intensive situation. And we made that in the rainy season and in the dry season. So it was a very tough experiment. What we saw was that those animals that have low delta hetal temperature, this delta, what means? We measure the temperature of the animals when they are in the climatic chamber, in the comfort temperature, and it's stabilized after 4 hours there. And then we measure the temperature when we submit them to a stressful temperature. Okay, and we compare the temperatures. So this is the delta, the difference between the temperature that the body temperature of the animal when the animal is in comfort and when the animal is under heat. So this is the delta, the difference of body temperature in both situations. What we saw was that the animals that had a lower capacity to variate its body temperature had much more homozygous genotypes coming from hosting. And the animals that had a higher delta, so a higher capacity to modulate its body temperature, had more genotypes coming from the gear breed. So what's more or less, let's say, the conclusion or the perspective about this study was that for the hosting animals, they do not have the capacity to modulate their body temperature as well as the gear animals does. So the gear animals or the Zebu animals, it seems that's easier for them to modulate the body temperature according to the temperature that I have outside. Okay? We have also done in the same population GWAS and post-GWAS study to identify candidate genes for milk yields in the Giroland. And also, we looked for the genotypes that come from hosting and the genotypes that come from gear. And what we saw about variance, I told you about genetic variance in gear. So we could see that, let's see just graph 1A and B. Graph 1 is when we consider, okay, the GWAS just for the effects of the alleles that come from hosting. And B is the effect when we consider only the alleles that come from gear. See that it's not the production, it's the variance, okay? So we can see that in the 4 main chromosomes for milk production, gear has a higher genetic variance in three of them, chromosome 1, 4, and 26, when we compare to the genetic variance that is provided by the hosting animals. So what it means for us, we are not talking about amount of production. We are saying that we still have room for selection, a lot of selection we can do in the gear. And we believe that this selection is not only considering the variance for milk production, it might be also for other traits, okay? And it's interesting because we can have which were the SNPs that we had inside the candidate genes that we found by GWAS. And we could see the genetic effect from gear and the genetic effect for hosting. And we could see the variance from hosting and the variance for gear, the genetic variance. Okay, so for example, for SNP1, it's almost the same. For SNP2, in the same gene, gear do not present any variation, but we have all the variation come from hosting. And in the third SNP, it's the other way around. We have a lot of variance come from gear and almost none from hosting, and so on. So it's quite interesting because we can see where the effects are coming from. So we saw that we had to keep working with gear and Girolando breeds. So one thing that we really had to work with was estimation of genetic parameters. We do not have too much estimation of genetic parameters. For example, something that is quite important for gear or Girolando is the production of oocytes and embryos, because it's really, really based in embryo transfer. Okay, in Brazil, we work a lot with these technologies. So we decided to work on that. And we saw also that we had to work with pedigree reconstruction to understand better the structure of the gear population using SNP markers. We could see, for example, that the effective population size is diminishing generation after generation. So in the last 5 generations, the number of the effective number of gear population is only 11. So nowadays, what the association, the Breeders Association, is doing, they're trying to get bulls from those herds that are not very used for milk production, that those lines that still a little bit, let's say, forgotten in the milk production programs, to bring them, okay, to increase the effective number of population and reduce our inbreeding coefficient. So this is the inbreeding coefficient based on runs of homozygosity. So we can see that we have a quite high level of homozygosity in the breed. So we actually need, let's say, to refresh the blood, refresh the blood of this breed, bringing more animals inside our pedigrees. We also studied runs of homozygosity chromosome by chromosome, and we could see that we have differences in the level of inbreeding according to different chromosomes. So we came back again to oocytes and embryos to make a correlation between runs of homozygosity, signatures of selection, and numbers of oocytes and embryos. And as we expected, according to the increase in inbreeding, we decrease the number of total oocytes, viable oocytes. And for embryos, we do not have too much impact, the level of inbreeding. But for the number of oocytes, we have a high impact on inbreeding. Inbreeding has a high degree of impact in the number of oocytes. And then we decided to check what happens chromosome by chromosome. And then we checked what is the percentage of runs of homozygosity and production of oocytes and embryos, chromosome by chromosome. Very frequently, of course, we have a decrease in the production of oocyte, even inside each chromosome. But for some chromosomes, we find that it makes not any difference, the level of inbreeding. So this chromosome, for example, doesn't impact embryo or oocyte production. And chromosome 14, we have a slight increase in the number of oocytes and oocytes, only oocytes. We didn't check for embryos here because we had no effects on embryos here. So it has a little bit increase in the production of embryo and oocytes according to the degree of inbreeding. Okay, but in general, we have a decrease in every chromosomes. We provided single-step analysis, something that is interesting. For example, here we are just working with runs of homozygosity and signatures of selection and looking for the areas that are conserved or not, or differentiate between animals that are lower embryo and lower oocyte production to those animals that are higher producers of embryos and higher producers of oocytes. Okay, so this is not a GWAS graph. Okay, this is a graph comparing FST between these two subpopulations. Okay, animals with a high EBV for embryo and oocyte production, and animals that has a low EBV for embryos and oocyte production. Okay, so this is the FST 0.15, and 0.15 means that everything that is above to 0.15 means that it's a higher differentiation between these two subgroups with high and low EBVs. Of course, I'm not going to show here the results for each one of these papers. You have the recording, you have the name and the reference. So those that are interested can go back and retrieve these papers. And then we made a GWAS study. People many times confuses the FST with GWAS. It's different. Here's actually the single step GWAS. and looking for genomic areas. We could see that for viable oocytes and total oocytes, we have the same windows that are different, almost all different for number of embryos. When we look to the Venn diagram, we can see that embryos has 42 genes that are individual for embryos. Viable oocytes and embryos share 28 genes. Total oocytes, embryos, and viable oocytes, they share 4 genes, 42 common genes between viable and total oocytes, and 1 gene is just individual to total oocytes. So in the papers, there is a lot of development and discussion about these genes. Okay. In this line, we still have 2 other papers, and the name of authors are all here. Okay, in general, there are students of mine that are doing their PhD or doing their masters. Okay, this one is under revision. There's a Journal of their Science, where we are developing daughter design to find out which are the best gear bulls to produce embryos and oocytes. I'm not going to show any results because we do not have time, and this data is still under publishing. Okay, and we are working in the parental origin analysis for the number of oocytes and embryo in gearcatus, trying to see if we have any indication of epigenetic effects in the genes that control the number of oocytes and embryos. So I'm also not showing any data because we're still working with this data. Okay, I hope we can have this published up to next year. But we do not only work with genomics, we have reproduction, physiology, nutrition, behavior, microbiology, and so on. This is a herd of gear cows. They are all donors, embryo donors. All of them are embryo donors. This was the first paper that we developed using maternal nutrition and genomics in Girolando animals. So this was like 2015. We could see that if we change the maternal nutrition from moderate to high during gestation, and we have males, we have also done this for females, but for males, we have no difference for the fetal weight, for reproductive organs fetal weight, testicular weight is the same. However, if we look to the seminiferous cord, interstitial seminiferous cord diameter, and seminiferous cord length, we're gonna see that we have impact on the maternal nutrition will impact the development of the testes of these animals. And we have also found this data for ovary development in the female fetus. So our question was always, I want to do that, but I want to take the animals to the adult age because I want to see if the maternal nutrition will impact in puberty and in reproductive life of these animals when they are adults, because that's okay. We already know the literature is full of example that if you change the mother nutrition during gestation, this will impact muscle. Okay, but I want to see what happens to reproduction. I want to see what happens to my mammary gland of this animal when they are adult. And it's quite difficult because it costs a lot for us to keep these animals under pasture, being fed. It's not easy. So we only could do that during pandemics. So this paper was published in 2015, and during pandemics, we could have a few Girolando animals. The association gave us the cows, so we could develop this research. And we also received for free the sexed semen for females, and we could develop this research. So we had 2 treatments for the mothers. Treatment 1, average daily gain of 600 grams per day, and treatment 2, the average daily gain of 300 grams per day during pregnancy. Okay, so the wafers were born, and after they were after weaning, we took these females and we began to evaluate them for the ovary development. And what we could see was that during pre-puberty, when the females began to have some development, the appearance of follicles in the ovaries, we could see that those heifers that came from the mothers from treatment 1, they had more follicles, ovarian follicles, than those heifers that came from the mothers from treatment 2. Okay, so in average here for treatment 2, we had 8 follicles, and for treatment 1, we have 15 follicles. Well, we measured all the biometry for the animals, and we could see that at pre-puberty, the withers weight and hump weight were higher for those heifers that came from the mothers from treatment 1. When compared to the heifers that came the mothers for treatment 2. And something that we were really interested on was the parenchyma. We could see that there is difference in the parenchyma when we analyze the image of the parenchyma for the heifers that came from treatment 1 and treatment 2. Also, treatment 1 shows much more parenchyma when compared to the heifers that came from the mothers from treatment 2. So yes, it seems that the treatment that we gave to the mothers affected reproduction and also mammary gland development. Okay. We are doing some genomic analysis in these heifers, but they are not done yet. And also nutritional analysis, but they are not done yet. This is ongoing, okay? So the number of follicles exactly at puberty for Girolando, we could see that group 1, there was not any difference between group 1 and group 2. For the age at puberty, so they reach the puberty at the same time. However, the heifers from treatment 1 present a higher number, a significant higher number of follicles in their ovaries. So if you want your heifer to be used as an oocyte donor for embryo transfer, you need to treat her mother well during gestation. So your heifer will have enough follicles in the ovaries to produce enough embryo for your embryo transfer program. Okay, so we're reaching our final considerations, and I can tell you that Zebu and the crossbreds are an open book for research. I came from pigs, as you, I don't know if you remember from my short biography that Kristen told you in the very beginning. I come from pigs, so I think a lot about crossbreds, and I always try to adapt something from pigs to the crossbreds here for the Girolando. And I'm a passionate person for Zebu, so it's an open area for research. And if you have time to see, almost in every publication that we have cooperation from a team that is outside UFV and many times outside Brazil. So we are always open to full cooperation with many different places. Okay, so genomics, reproduction, physiology, nutrition, microbiology, behavior, it's a lot of things that can be done. And I think that we actually need to disentangle which are the Zebu opportunities to increase production in tropical and subtropical regions in the globe. So I think that Zebu and crossbreds might be a solution, for example, for the poor areas where they need to produce milk. For our team, our future perspectives, we need to deep sequence all these QTL peaks that I showed you for tick resistance, heat tolerance, milk production, oocyte, embryos. We have to open these areas and understand what do we have there inside, which are actually the quantitative trait nucleotides. If we have regulations such as long no coding RNAs, or if the regions are controlled by microRNAs. So we actually need to work on these genomic areas. It's an open source of research as well. Development and applications of models to study dominance, epistasia, and pleiotropy. Another student of mine, I have seen him here at the very beginning, Mateus. Mateus, are you still there? And Mateus, his postdoc, his PhD will be probably working with epistasia and pleiotropy in the Girolando breed, because we also need to understand better the effects of these effects, especially when we saw that we have different effects depending on which side of the pedigree our alleles come. We are still collecting more phenotypes and other different and intermediate phenotypes. For example, number of embryos, number of oocytes is an intermediate phenotype to number of heifers that are born. So we need to keep working, disentangling these phenotypes. Genotypes by environment interaction study is, of course, very, very important. Using random regression models, for example, we need to keep on this track, okay? So if you have any questions, any comments, one more time, I need to tell you, thank you very much for the opportunity to show a little bit about what we are doing here in our team. And I really thank you to my students, to my cooperators, because of course, we had a lot of help to do all the things that we are doing. Okay, thank you. Christine, are you still there?
Yes, thank you. If anyone has any questions, yeah, you can raise your hand, or if you want, you can also enter your question into the chat as well. But I could get started on a question. For when you were looking at fertility traits, it was interesting that some chromosomes had an increase in oocytes when there was also increased inbreeding. So does that have any maybe practical implications to how we maybe assess inbreeding or something else?
Well, actually, yes. So these areas are also in our list for deep sequencing, for we better understand what's going on in those areas, okay? And it might be that the genes that we have inside these areas, they are not deleterious, and they have just effects, okay? But they're not deleterious, so they just are going to produce more oocytes, and nothing deleterious produced by them. This is a hypothesis, but we actually need to know which are the genes that are there inside. And what we have overall, what we have is that inbreeding will decrease fertility, because if you look over all the genome, the pattern is that inbreeding will negatively impact fertility, despite one or other region that will increase it, okay? Yeah, it's tricky, but it's nice.
Yeah. Yeah, I thought it was interesting. And then Samla, go ahead.
Yeah, thank you very much. Thank you very much for your talk. It was pretty good. I was wondering if you're talking about the hips from cows that had different diets, and I was wondering if this would affect also the male sons of these cows with different levels of dietary.
The sons.
Yeah.
Yeah. We would love to work with the boys as well, but we used sexed semen, okay, because we had limited resources, a limited number of cows that were donated for us, okay, and we would not have room to develop experiment for the heifers, another for the young bulls. You see, so we used sexed semen and had just heifers. Okay, and of course these heifers, they are in our herd, and as a milk herd, they are of interest because they will stay here as cows. They will deliver, they will produce milk. For males, the economic impact of you have like 10, 12 males being born, you know, in our dairy station, you know, it should be even more expensive. But I have a lot of interest in that because we know that spermatogenesis is much more quantitative than oogenesis. So my question for males is still there. Will the maternal nutrition impact the amount of spermatozoa that a male can produce? Because they produce billions of sperm. It might be that it doesn't impact, you know? But it's still there, the question.
I see. Yeah. Thank you very much for your answer and for the presentation.
Okay.
And we have a question in the chat. We know that Zebu is more resilient and robust. How can we apply the knowledge acquired from these studies with Zebu to select animals more resilient?
Okay, you mean you want to apply this knowledge with Zebu to select animals more resilient, but you mean select Zebu or taurine animals? Leticia. Well, in taurus. The point is that the intricate genetic regulation that might be involved in these traits. You see, first of all, we need to check if the alleles that are present in Indicus, if they are there also in Taurus, because it might be that they're just not there. The genetic distance between Taurus and Indicus is quite large. Okay, it's quite large, so it might be that the alleles are not there. For example, the 700K SNP chip, you use around 400K SNPs for Zebu. Okay, so we think the other way around. It might be possible that a lot of alleles that we find in Zebu are not there in Taurus, first of all. Second, if they are there, we need to check the effects. Something that is quite nice to work with crossbred is that you have the genome of both breeds in the same animals and do what we have done. And then we have the genome of both breeds inside one animal, so we can check the relationship between both genomes. Okay, as we have done, and I showed you for Girolando, and many times the control that we have for Taurus will be different for the control that we're gonna have for Taurus. For example, we are working with microRNA. I'm sorry, another example from pigs. We are working with microRNA in the development of embryos in pigs, and we have seen that some microRNAs that help in commercial line of pigs to develop myogenesis are not present in those fat type pigs. Okay, so it's a good question. It is a good question, but I think we need to disentangle a little bit more the genome. Okay, Leticia.
Great, thank you. And it looks like we're out of time. So thank you everyone that joined us today, and thank you very much, Dr. Guimaraes, for the very great presentation. See you everyone next week, and everyone have a nice weekend. Okay, I say thank you for you, all of you. Thank you very much for the space. Valmir, see you. Valmir used it to work with me here, and now he's in Canada.